I'll go ahead and introduce myself. My name is Carol Thompson. I'm at the Allen Institute for Brain Science. I am part of the Brain Initiative Cell Census Network at the Allen Institute. We have the Brain Cell Data Center, which is serving as an organizing hub for the Brain Initiative Cell Census Network. In this consortium, we have a federation of data archives for different data modalities, and the BCDC, we host the portal for the consortium, as well as a documentation of all the data sets that are available in the consortium and where they're available. And we're moving towards making that a more searchable registry of individual data. We're interested for sure in understanding how we interface with all the different metadata standards efforts that are either have been done or are currently in development. There are a lot of new efforts that are being funded by the NIH at this time. And as I understand it, if we can find ways to make all of our metadata more standardized across different consortia, this should help us find data, help people reuse data and analyze data, and make things more fair at the end of the day. So I think it would be great if we would start off the session by everybody introducing themselves, your name, where you're at, what consortium or consortia that you are involved in, and perhaps give a quick statement of what are some of the pain points in this area that you are feeling and would like to address today briefly. So why don't I pick on someone whose mic is off? Heather.
So that's perfect following upon Carol. I'm also a part of the Brain Initiative Cell Census Network. I represent the NEMO archive, or the Neuromolecular Omics archive for the BICCN. And Carol and I have worked quite a bit together on establishing metadata standards for that project. I'm also part of the Common Fund Data Ecosystem project, which is looking to establish a consistent set of metadata standards and a consistent metadata schema across a number of projects. And so in the last session that I did this morning, we talked a lot about one of the difficulties being lack of clarity on use cases. And I think that's one of our biggest obstacles, is really understanding what metadata we need in order to address the use cases of our clients.
So I had forgotten to ask in the beginning for scribes, volunteers to be scribes. And I think while we're doing this quick introductions, we're actually going to come up with some things that we would want to put up here. Can I have 2 volunteers to be scribes in the Google Doc?
Hey, this is Kylee Degatano, and I'm here to scribe.
Wonderful.
I'm happy to try to help.
Okay, wonderful. Okay, Jason, why don't you go next?
So my name is Jason Swedlow. I'm at the University of Dundee in Scotland. I run the Open Microscopy Environment project, or OME. We build open source software and specifications for imaging data across many different modalities, across a lot of biological imaging and biomedical imaging. And my group has built the Image Data Resource, which is a public bioimaging data publication systems running at EMBL API. Pain points, the ones that are sort of crushing us on a daily basis now, are metadata specifications for bioimaging data sets to be published. So papers are coming out. How do we get the metadata specifications and the tooling, in particular, into the laboratories that are generating these data sets, so that they can submit their data sets and, I guess, as Carol said, meet all of the criteria for fairness and so on. Stop there.
Thank you. How about Mark?
Hi, I'm Mark Musin from Stanford, and I'm kind of an interloper here. I'm not currently funded by the Common Fund, but a colleague who is asked me to join this meeting thinking that I might be able to contribute. So I work in the area of biomedical ontology. We manage at Stanford Bioportal, which is the world's most comprehensive collection of biomedical ontologies. Something in there might be useful here. But more important, we also manage CEDAR, which is the Center for Expanded Data Annotation and Retrieval, which basically is web-based technology, which I think may answer some of Jason's problems. We provide mechanisms for making it possible to have libraries of metadata schemas and templates that people can share. We can use those templates to create metadata that are annotated with ontology terms and other appropriate data, and then upload the data and the metadata to corresponding repositories. And we have a number of collaborators who work with us in this area, none of them actually in this particular consortium, so maybe there's something we can talk about.
Okay, wonderful. Okay, how about Benjamin?
Hi, guys can call me Ben. I run the ENCODE Data Coordination Center. We have been, I would say specifically regard to this meeting, I believe we have one of the most extensive metadata systems for any sort of public genomics project. I did a census the other day. We collect something like 97 different objects in JSON LD and JSON schema. We have over 1,000 metadata fields. I think we're happy with most of our system. We've sort of developed it over, I mean, something like 7 years of interactive work with the labs. We learned a lot from previous iterations of the ENCODE and the ModENCODE data coordination centers, what they did well and what they didn't do well. I think we've done a pretty good job of like explaining to the world what, like how we do things and why we think it's important, but I'm just here to like help out, like let you know what we think is good, what doesn't work.
Okay, great. Okay, how about Jessica?
I am Jessica Langer. I work for the Helmsley Charitable Trust. Excuse me, we are funding an initiative called the Gut Cell Atlas, which really should be called the Crohn's Disease Atlas. We're doing a normal and healthy tissue compared to Crohn's disease patients. And I'm here because for us, the real challenge is because we are so exquisitely focused on Crohn's disease is how to get the Crohn's disease, the patient metadata correctly into a database in a way that we'll be able to get insights into disease progression and physiology.
Okay. And what is your, I was going to just ask that, what is your challenge there with the patient metadata into?
It's basically, we want to be able to take our findings and bring it back to the patient and the type of disease that they have. So we have to all agree on how to, best describe the patient, their symptoms, where they are in the disease, etc., and medication, okay. Things like that.
Okay. Ingrid?
Hello, I work with Ben at the Data Coordination Center for ENCODE at Stanford. Really just listening in to understand some of the other systems for metadata coordination and how standards are developed. So thanks for opening this up to people.
Wonderful. Thanks for joining us. Kylee?
Hello, my name is Kylee Degatano. I work with the Broad Institute on computational pipelines, and I'm part of the Human Cell Atlas project and the BRAIN initiative. And just here to take notes, but also very happy to listen to anything that helps successfully set standards.
Great. Okay, so what we'd like to do in this part one session is collect how might we. So when we're doing some of these things, we want to find our problems, and this is basically coming up with our use cases for this part two session. So part two is problem solving. Part one is identifying the problem space that we want to address. So some of the things that we were saying in this first part of the session could potentially start being turned into some of these how might we. So that's what the HMW stands for in the Google Doc. So as we go along, if our scribes could try to formulate some of these as how might we's up here in the top, that would be great. And you can just use the HMW for the how might we. So for instance, part of the problem that I was seeing is when you've got a consortium either a brand new consortium or one that's still working on maturing its processes and its metadata. One of the problems that I see, and I'm going to start here and then we can open up for discussion on clarifying the problem space without trying to solve anything yet in the session. But one of the problems that I have is really trying to understand what are the standards people are using, what are the standards different consortia are using. So there are many standards out there. I can go to NIF and I can search for standards and I get back so many different things. And it's kind of hard to actually bring this together and figure out what are people actually using, what are the problems with it. There may be standards, you bring them in, you have to adapt them. What kind of adaptations are people doing? So one of my big ones is just how might we enable clarity on what different standards are currently in use across different consortia. I will say one of my problems is I don't even know what all the consortia are that are out there. So having being able to figure out what is the set of consortia that we really ought to be easily aligning with is also a place where some clarity could be used. And I'm going to open that up. Does anybody want to comment on that one or bring up another how might we?
Well, if you're looking for comments, I'll jump in. A couple of things come to mind. One of which is a resource out of Susanna Sansoni's shop at Oxford called fairsharing.org, which purports to be not a repository, but a registry of standards that people use. It has a lot of play in Europe, doesn't necessarily have all of the level of detail that I think the consortia here need, but it's a resource that I think is valuable. The other thing I will say, with all modesty, is that we could actually easily set up a set of libraries within the CEDAR platform, which would allow members of the consortium to create templates describing their metadata schemas and be able to have them all in one location, like a Google Doc, that they could then compare.
And could you just spend another minute and talk? I know you did in your introduction, talked a little bit about how you can make these templates. What kind of coverage do you have right now for what templates you're currently making?
Well, we're totally agnostic about what the templates actually contain in terms of content. So what we provide is a mechanism where we allow people to author templates by describing the kinds of fields that need to appear in the metadata. What are the types of ontologies or branches of ontologies that should be used to provide values for those fields. We also support the common data elements that NCI uses for its CRFs, and those also are available as metadata elements. And so we are open to any kind of content. The main advantage of the CEDAR platform is to provide some kind of standardization over the names of fields and the kinds of data values that are used for those fields.
Does anybody else have any other comments on how they gain clarity?
One of the tricky things about this whole problem that I'm not sure everybody understands is that one of the difficulties in writing standards ahead of time is that you can only mint a set of standards after after all the experiments are effectively, I don't know, necessarily done, but at least, slotted, right? So if you're in a functioning, like in our case, if you're in a functioning environment where people are, coming up with experiments and submitting experiments to you, like your metadata model has to change, right? It's not enough to go to, and either that or it's so simplistic that it doesn't actually solve any of these issues. So I don't know how to federate that across 20 systems or however many people are on this call. I guess it's only 8, but and Ingrid's in my group, so it's only, I don't know how many different consortium need to do this, but you sort of need to break down the problem into what's static and what's dynamic and how to handle it.
I guess I would comment on that. Part of your statement was about, you can't mint the standard until the data's been collected. But on the other hand, you can't collect some of the data, metadata or set standards for some of that after the fact, because then people aren't going to collect the right things, especially when you're in a consortium, right? So you have to.
I guess I should clarify for sure there. It's the difference between what you collect and what the standard is. Like you have to collect more than what the standard is. You can't. So you have to. That's what I'm trying to get at, is that you won't know until the project is done what the necessary standards were going to be.
But you know what the floor is. You know what the minimal kinds of data are. If you
set the floor, you'll get the floor. Right? So that's the impact that I'm trying to make, is that you can set a floor as this is absolutely the minimum stuff you need to do. Like, for example, if you were curating experiments out of Geo or something like that, right? You know what's in Geo, you have a floor, right? There's no way you're ever getting any more information out of someone who's written a paper and stuck it in Geo or whatever. But if you're in an active consortium, like, and it depends on I can only speak from our experience where, in a 4-year granting period, people start the 4 years saying, I'm going to do these experiments, but 2.5 years in, they're actually already changing their mind to what experiments they're actually doing. And sometimes those experiments will add whole new chunks of metadata area that you didn't even think about 3 years before. So that's where this is the difference between collecting metadata and actually standardizing metadata, right? You have to collect more than you can standardize. You can't, because it can't go the other way. Does that sort of make sense? Maybe I'm just sort of.
No, it does make sense, but I think the question then becomes, what, let's not get into too much semantics, but what is an experiment? If you can identify up front the kinds of investigations you're doing, what kinds of instruments might be involved, what the data might look like, all of that is actually not yes, it's a floor, but it's not so terrible. And as you understand more about what additional annotations you may want to make, that kind of stuff can easily come later. I think the question is, what kinds of things you need to say about the data so that the data becomes searchable? Because the things, I know we'll get to a conversation about fairness later, but without being able to have a statement up front about what the minimal data elements are going to be, what the minimal metadata elements are going to be, then search becomes really problematic.
Yeah, I agree. I mean, as part of the NEMO archive portal, you have to start somewhere. And so we've started with sort of that, I mean, it's an expanded floor, but that basic metadata that can be standardized, but we're fully aware that there's going to be other areas of, additional metadata that we're going to need to capture that are more dependent on the specific research studies.
Yeah, I think we're all saying the same thing, right? Yeah, I'm not saying there can't be a floor. Is it there has to be? I mean, I don't think I'm being super clear, but that maybe I'm just thinking about it from the perspective of having to develop a metadata model while there were no standards at all, right? So in 2012, there were no genomics metadata standards at all. Like, we inferred our standards based on what was done in Roadmap and ENCODE from like 2008 to 2012, right? And we realized that back then that the standard that we were like reverse minting out of what had been done, like, was never going to be adequate for future experiments. So we came up with a system that handled that, but then is extendable. Right. So is that really a floor? Like, so what, I mean, because you can set a floor, but what happens, like, I mean, as a specific example, a few years ago, people started submitting to ENCODE these experiments where they would start knocking down genes in cell lines and doing RNA-seq, right? So there's some shRNA, some siRNA, and then CRISPR, right? So, and this wasn't, like, people don't submit to you at the beginning of the granting period, what are all the experiments they're going to do necessarily, or they didn't to us. If they do to you, that's great. So, but we didn't even have a system for storing what happens when you take a cell line. Like, I mean, ENCODE's weird. It doesn't, we have a lot of tissue data, but we have a lot of other data too. And so when someone starts creating ideas that you didn't have, you know, you have to then have a way of extending the standard further, I guess. That's all I'm trying to say. Maybe that's not helpful federating though.
I rephrase this a little bit as a how might we set standards before experiments are done versus improve the standard after data is collected, or how might we set standards for new technologies? I feel like that's part of what you were talking about, some of the challenges you had when you had newish technologies or before there's an official standard set. Does that capture it somewhat?
Yeah, I think that's just my, I just feel like it's a concern that whatever federation standards the super consortia come up with, that they are prepared to extend them, right? So, it just means you just have to talk a lot, I guess. I don't know what to say. I think you're all right.
So I would ask everyone, how, if there's someone who'd like to comment, how are you finding standards, and to what degree are the standards you're finding to use for your purposes requiring adaptation? Jason?
As a first question, you call your friends. And you do what they tell you to do, largely. And that seems a little bit flippant, but actually, so in our experience with trying to build the image data resource, we had a lot of annotations to add, phenotypic annotations, et cetera, around what things that, so we're receiving data sets from a variety of groups to be published. We have phenotypic statements that those submitters are making. They don't have the tooling to translate those phenotypes or measurements or whatever to some sort of controlled vocabulary. We call our colleagues in various places, say, help us. They give us directions, suggestions. We come back with feedback about what works and what doesn't, et cetera. And we were talking about this in the last session, so I guess in particular for Carol and Kylee, this will be a bit of a replay, but some combination of the kinds of resources that were mentioned, as Mark mentioned, NCO is amazing. But on the other hand, I guess, as Ben is sort of saying, mapping real experiments to define concepts, there's always a bit of a crash there. And so, yeah, so call friends, do what they tell you, identify the faults, and then contact the people who can help you fix them, I guess is probably the best answer that I have. But that's from someone who doesn't, I know nothing about developing these things. I'm just trying to use them effectively and be a consumer of them. Yeah, that's our experience.
So it's always, the standards are only as good as their use, right? And so this is the one thing where everybody's trying to set a standard. There's standards that are being set outside of consortia that are absent in implementation. Then there are standards that are effectively being set because they are being set for a group of people in a consortium. So this is why I'm curious, like when we look at external standards, what people have been able to apply wholesale, or where is the mismatch going between standards that have been set versus the ones that are being implemented in the consortia, right? Or is there a difference?
I think that's a really good point, Carol, and I think that's maybe where I was getting confused. That everybody gonna have their own internal representation of their metadata and data, and that probably can't be standardized, right? I mean, maybe at the level of like HCA subgroups or something like that, I mean, if you're not sharing software, then it's going to be very, very difficult to do that. But I mean, and I guess some of you were on I can't remember all the names of these calls, but the earlier session on sort of data on like data sharing across consortia. Is anyone here on that call? Maybe. Like I think what we would have to do is we actually have to, there has to be some set of like, I guess IHEC did this type of thing where they shared a way of describing genomics tracks so that you could like display them together effectively. Like this is kind of, or GA4GH has this sort of RNA get system of doing things. And so you have like substandards of data sharing in some sense, but you need a way of defining what possible things could be shared. Like it's a very large, the problem with these projects is they're very large, like these standardization projects can be very, very large, take a long time, and they don't tend to be well funded. Like there aren't people who are set out, like you would need, I mean, you can't really have half a person per group doing it, right? Like that's millions of dollars.
If I could just try another answer to this question, I can tell you in the world of imaging, how do people, quote, find standards? By and large, they end up adopting software that exercises those standards for them. And so in most cases, and here what I'm referring to is less about, for example, a specific part of HubMap or Brain or HTAN or whatever, but just in a much global sense. People use standards all the time without actually, in most cases, without even knowing that they're doing so, without explicitly saying, I'm going to adopt this. There's a piece of software that either is embedded in as a library or whatever, and they may or may not even realize it, but that is utilizing that standard and providing a way of exercising that standard. So I think when it's kind of, I'll apologize, Carol, and you can tell me to shut up, but rewording your question slightly as opposed to how are you finding standards, which I actually with this group is actually probably an important question, but also how does the wider community find these things? They end up with a tool that works for them. I suspect that's at least in the imaging world where I know something that's probably most common there.
I think that's a really great point, Jason. I think it's actually really, really important in that in the genomics world, and that when I was talking about IHEC standards, we were talking about essentially genome browser mapping, right? And this is just, there's only two different types of files. You're just essentially labeling the tracks in a way that is roughly consistent across 10 consortia. And so this is mostly histone ChIP-seq data, like it depends on whatever your data type you're really working on. And for these more, but that's actually, and it actually naturally works that way. Like if there's a standardized software for interacting with whatever data we're interacting with, right? Single cell RNA-seq data, for example, I think is one of the reasons like a lot of us are here. Then internally, like that software has a particular standard, and if people use that software, then our groups individually have to effectively export our data into something that is consumed correctly by that. I don't know what those tools are in the future tools, but that seems like a really good way to do it. Of course, the problem with software is that unless somebody Microsofts us up or whatever, right? Like, like whose software wins?
Mm-hmm. I rephrase that in here as how might we integrate with standards set by software or data acquisition platforms is another one. So the little notice came out that we have 5 minutes left. Other things that we could have addressed in the session would be how mature are the standards that are out there? What additional metadata standards are needed? I don't think we're going to be able to address specific metadata standards for anything. There was already the clinical metadata session earlier today. But perhaps we could talk, the other things that I have on the list are what are best practices for metadata standards development, implementation, and documentation. And then there's a piece on ontologies. I think I would skip that at this point. So with these in mind, if we want to spend part 2 addressing solutions to something, if you scroll up a little bit, we have some how might we's, and Kylee, do you have any more how might we's in these notes down here?
Probably. Let me take a look.
So the ones that I had written up at the top were how might we enable clarity on what standards are in use in different consortia. And from the early conversation we had, it sounded like there were some ideas already on things we might be able to do there. How might we set standards before experiments are done? Or how might we set standards for new technologies? Part of this might get to how might we develop a common best practices for setting standards. I think it kind of gets back to that. We're all trying to do this on our own, and as new consortia are coming online, this is just my perspective here, having a playbook for how you're setting standards would be awfully nice, and how you reach out to looking at existing standards, how you map to them, how you develop new ones. And then how might we integrate with standards set by software or data acquisition platforms? So, and then there's a how might we define common metadata concepts and standards that have the most value. Does anybody have any specific feelings about which ones we want to concentrate on in part 2? Heather, I'm going to pick on you this time.
Well, that's not fair because you've already picked on me. I don't know. I was thinking that last one, setting a playbook for setting standards, finding them, integrating them, sort of encompasses some of the other how might we's. So that might be a good thing to focus on.
Okay.
Might want to grab Marianne Martin, who's, I think a lot of what, if I understand correctly, Carol, you'll know better, that's an awful lot of what she's doing.
Setting a standard for setting standards. I asked her, I'm like, do you know of, or have you even written an article about how do you set a standard? And she said she doesn't know of one. So this is a place where we actually, I feel like there's a lack. If anybody knew of such a thing, she would know of it. They don't have
exactly.
it.
That's what it is.
Right now in the Google Doc, we have for the breakout session part 2, there's a space to have additional notes or recommendations or ideas before we get to the final template, which is where we'll put the final plan of how we would want to follow up, recommendations of what we would want to do and what additional expertise we may need. But I'm hoping, let's see, we've got Mark, Ingrid, Kylee, and Anita. I'm sorry, you weren't here for our initial introductions. I'm Carol Thompson. I'm from the Allen Institute for Brain Science, and I'm part of the Brain Initiative Cell Census Network. Would you mind introducing yourself really quickly? Okay, I can't hear Anita.
Hi, hello.
Hi.
Hi.
Would you be able to introduce yourself very briefly for this group?
Yes, I am a, let's see. Right now I work as a consultant for in helping people standardize clinical data, I guess, across data sets for the purpose of performing aggregate cohort level analytics. Let's put it that way.
Okay, thank you.
Yeah.
And are you involved with one of the current consortia funded by NIH?
No, I'm not.
Okay. Okay, well, let's go ahead and get started right now. I don't see Benjamin back yet, but maybe we can go ahead and get started. So we decided that we were going to work on setting a playbook for setting standards, finding them, integrating with them. We might break that down to how do we help people find standards, and then what are best practices for setting standards. So I think this is where we can start thinking about ideas for solutions. Part of those may be things that we can do. Part of this may be things that we need other people to help with or to do. So we can have recommendations of either sort in this session. So I guess I would, let's see, to start with this, would it be okay if we started with how do consortia find standards? We can go ahead and put in the phone, your friends, and then, as Jason had suggested, but then if we each have a consortia and we have standards, maybe we can talk about how we're putting our standards out there, and then we can talk a little bit more about how we best practices for setting standards. I feel like it's always good to find standards that you can start with because somebody else has done a lot of work already in many of these areas, and making sure that there's not a standard out there that already exists could shorten your time to developing a new standard in whatever area.
I'd love to hear Bart tell me how I should do it as opposed to how I'm screwing up.
Okay, so for finding standards, if we start putting in some of these examples of things we could do, there are different registries for standards, like there is the fairsharing.org. You can check there, although again, it's hard to know who's actually using these standards. There's a lot of standards out there, and it would be lovely if you had in there, and this standard isn't used by these groups or this consortium or this software, right? So understanding who's using it for any one thing. So some of this could be feedback we give to these places that are logging standards to make sure that this is information that is collected on who is using these standards.
Yeah, I mean, from an omics point of view, I know that that's something that the Genome Standards Consortium does. Like on their site, they have different projects that are using their standards. Similarly, disease ontology, they definitely publicize who is using their standard. But I agree, I think that's really useful.
And then for any given consortium, I'm assuming if they have standards that they are using, like you said, we should all be putting those standards out as well.
Yeah,
I feel like, go ahead.
They should probably be, on the, both like in sort of a vaguely human readable format on the web, and also ideally discoverable. Like you can discover them. They're in a format like JSON LD. You can like look them up somehow. I mean, they're pretty wide ranging, so it's kind of hard to figure out what the right question to ask is, but I think a lot of them are there. But your point about not knowing who all the consortia are is kind of a tricky problem. I guess we could have a wiki page. I don't know.
Who are the consortia? You need to know that before, and what generally they're working on that is relevant to you if you're trying to find similar consortia to harmonize with.
Can I step out of line here? So as a European, and I've gotten myself into, I think, a little bit of trouble. So I guess I'm an external program consultant on the HubMap project. And it just seems to me, and I know people in H10, and I know people in 4D nucleome, and I think there's a couple others as well. And it just seems, and what I can't get, I just can't figure out, is how all of these different entities, and Kel and I know each other from work on a little bit of brain metadata. All of these different entities, all nominally funded by the American taxpayer, all doing nominally, obviously not really, but working on the same thing. But even just to say, just to address these questions, what are these consortia? As Carol said, you actually have to know what the consortia are, first of all, and then who's actually doing what. As I was sitting here listening to this, I was thinking, why isn't everybody just pulling in NCBO or effectively, so there's each one of them has a DCC or a DCIC or whatever they call it. And, so is there a way, Mark, I guess it's a fairly directed, pointed question, but are they pulling in your resources? And if not, like, there's nobody at NIH on this call, I guess, but it's from the outside looking in, it seems really hard to kind of understand why it's so fractured.
And
Mark,
it doesn't have that kind of control over its grantees. It just doesn't.
Mark, you're on mute, Mark.
Sorry about that. Obviously a loaded question, Jason, but
sorry.
At the level of ontology, certainly BioPortal gets almost all the traffic, and people know that they can find the ontologies that people are using. And frankly, as people argued earlier, we make sure that our metadata makes it clear what projects use those ontologies, and people can see summary statistics that tell them how often those ontologies actually get used. So I think from that perspective, we have things covered. Making metadata available in a public way for people to know who's using what is hard, and that's more difficult. You go to fair sharing, and what you find there are not really the metadata that people are using, but you find the minimal information checklists that are associated with different experimental types, and Miami being the poster child. But going from such a checklist to something that's actually usable is a big distance. And frankly, what we have done in our group, since we don't actually make the standards ourselves, but we work with collaborators who do, is we can be a kind of broker to let them know what standards are out there, what people have used, and help them as they create their own. Because basically, we've never worked with any consortium that has been able to take a standard and just use it off the shelf. Everybody wants some tinkering. And I think the most important thing that we can do is to make sure that the key fields are standardized across various use cases so that we can make sure that they all become queryable in the same way. But I don't have any expectation that anything is going to can be used en bloc.
So there's a grant as well that was given by the NIH to RTI International and PI Carol Hamilton, who's leading a standards development effort that Jason and I were part of. And they have done this sort of effort for, I believe it was phenotyping was a big one they had done before, where all the standards end up getting logged onto a central site. And so they have a grant to do some of this, but I think it's restricted to brain, whereas this gets all confusing because if you're working on lung, we should still have the same standards for the same data modalities. If you're doing spatial transcriptomics in lung or in liver or in kidney, it should be similar to what we're using in brain.
Yeah.
Transcriptomics or epigenomics, we need similar standards. So you can break things out by data modality, but then you have consortia. This is where it's just trying to pull all these things together. There's a lot of different registries, and having one place where you can just find everything would be great. So CEDAR is a place that's logging some of the, is this a place where everybody could log the standards they're using and their deviations from a standard? I know you said that you had the metadata templates in there and you had the ontologies.
Yeah, so it would be very doable to create spaces within CEDAR, and CEDAR allows you to have private spaces and publicly shared spaces where the various groups could each identify what their particular metadata standards are, put them in a library, have that library shared with other members of the larger consortium. And that would be one way of at least knowing who's doing what. It doesn't address this for the biomedical community at large, but that's actually something, with funding, we could provide the same kind of capabilities within CEDAR that we do in BioPortal that would be able to log usage and provide some guidance in terms of what people around the world are doing with these various resources, but that's not something we currently can support.
So it's late here, and I'm starting to get a little tired and punchy, so I'll just ask the direct question. I mean, in terms of making this playbook, I guess you're trying to ask the direct question. again, Carol, tell me if I'm off topic here. But if we tested that concept, Mark, so if I got my team to try to effectively, represent what we're doing in CEDAR and others around, other consortia around the table, just to effectively test that concept, is that useful?
Yeah, that's a defeasible hypothesis, and we could actually try it out and see if it works. I actually think it could be effective, because if people knew that they were all going to the same library, and that within that library were collections that were used by various consortia, you could at least have the ability easily to look and compare and understand how your standard could actually import someone else's standard and build on it, rather than having to start from scratch. So I think it's very doable.
Okay, so there's at least CEDAR is one mechanism that could be looked at for ways that consortia can publish and compare metadata schemas and standards, and there may be others. I would think as a next step for something like this, it might be interesting to get NIH to help bring people together and bring CEDAR, maybe the work that Carol Hamilton's doing at RTI, and I don't know if there are some other other representatives of some of these things that we could have a couple talks by, and then I feel like a bigger brainstorming around kind of how we might be able to leverage some of these tools to share data standards, to search and share these standards would be a useful follow-up.
Yeah, the other thing I will mention is that in Europe, the GoFair initiative, which is funded through the European Open Science Cloud, has gotten religion about standardized metadata. And actually, they have a lot of religion about CEDAR. And they're starting to have what they're calling M for M's, metadata for machine workshops. Or rather, they're starting to plan them. They actually haven't had one yet. But the idea would be to use CEDAR as an authoring platform that would assure that standards for metadata are consonant with existing standards, that they use ontologies and appropriate controlled terms appropriately. But these would be workshops that GoFair would basically run. Exactly who would pay for them, I'm not exactly sure yet. But the idea is they really want to start creating metadata that would be actionable, that would be JSON LD compatible, and all the good buzzwords. And maybe there's a mechanism to try to piggyback on activities at GoFair that would allow us to create workshops, when we have workshops again, that would allow us to be able to do this kind of work.
So I'm wondering, from the perspective of our group today, if we should put all this in the bucket of participate in a workshop, get NIH consortia specifically in a workshop around finding and publishing standards, but we could also look at how this might interact with the M4M and the GoFair and CEDAR. But I think it could be a workshop with like a number of different groups that we could have.
Right, so CEDAR provides a technology platform that would allow creation of libraries and comparisons and being able to utilize other people's standards and perhaps repurpose them. The M4M workshops are going to be a series of activities that would, for various groups of investigators, come up with standardized metadata. And frankly, if someone else is paying for them, then I would take advantage of them. The bigger question, what are the right standard operating procedures for making metadata standards? That's a harder question, because that's like, how do you make sausage? Even if Marianne were here, I'm not sure she would tell us. It's a very hard political process, which really requires ironing out differences among people who have very strong opinions about things that often don't matter that much.
Mm-hmm. Are there any things for the best practices for setting standards within a consortium? Are there things that we can agree on? I mean, I feel like the first thing is find existing standards and understand where they fall short for your consortium before you make a new one, ideally, right?
Yeah, exactly.
Of course, a lot of times that all takes a lot of time to sit in and dig in on that. So you get started with something, and then we continue to look at these other standards and understand from a practical standpoint, how these are implementable in our consortia. Is there anything else that we could bring up for best practices, or maybe it's not what the best practices are right now, but how do we get to a place of understanding what the best practices are?
I'm sorry, can I ask a quick question as to
Sure.
when everyone here is talking about metadata, what kind of metadata are we talking about?
So I think this could be, I mean, metadata in many ways could be seen actually as data as well.
Exactly, that's why.
It could be data set metadata, it could be provenance metadata, it could be information about the analysis that was run, it could be, at some point, it could be results, how you're putting results of that analysis into a standardized form.
Yeah, thanks. I just wanted to make sure I'm understanding it correctly as everybody else is.
I see this largely as the big problem we have in the consortia. If we're talking about how do we enable collaboration across consortia, how do we enable data analysis across consortia, it's about any of these things, whether it's the provenance or some of the results at the end. How do we put them into a format? How are we setting standards for how this information is collected and provided to the community? Okay. Any other thoughts on either what are best practices for setting standards within a consortium, or what would encourage people to integrate with current standards?
Carol, two things, and again, this is very biased based on the experience that we've had in imaging, but it's probably just one word, well, implementation of some sort. So some kind of implementation, mindful that rarely do we have absolutely fully developed, completely mature tooling, et cetera. So that either is some library that implements the software that can, implements the standard that can be used, or, I guess it's or, some repository or resource that is implementing that standard that effectively is driving it, which is possibly saying two ways of saying the same thing. It just kind of depends. But
you're saying that a standard should be set by the use cases and actual implementation need.
Yes.
I agree with that.
Yeah, I definitely agree with that too. I mean, I think it's hard to do it top down, right? It's just hard to do it top down. I guess if I was going to add something to the playbook, like rule 0 should be like, and maybe we all know this already, but like use ontologies, do not use plain text description ever. Like ever. I mean, even if you don't,
except where you need the prose that describes the experiment or something. Occasionally you need a friendly paragraph on a landing page.
I mean, you can have that. I mean, right, but it's a, well, what we actually do is we actually compose sentences sort of like based on it. Like, I mean, yeah, I mean, it's not that you can't store it. You just, it has to be only redundant information. Actually, it's, because the computer will never parse that, so.
Okay, how about anything else for setting standards? I'm going to add in here the check existing standards first, which is why we need the solution to part 1. And I guess, you know, I have big questions about how do you decide when you should deviate and when you shouldn't, right, from existing standards. Usually it's just some implementation piece on your end, and then maybe it really is just you need to provide a mapping to an existing standard or to other standards. Okay, let me open this up and ask, is anything else? But what can we do to move this along? Is there something we feel we can do? I mean, we already talked about it would be nice to have some sort of workshop in the NIH space to talk about how we can make it easier to find and share standards that are in use by the different consortia. What else can we do in the space of setting or identifying best practices for setting standards? Is this something where this is more of a polling different efforts, getting retrospectives from different consortia and understanding what worked, what didn't work, what pitfalls they had, or how can we make this move forward?
Well, I'll bite. And what I will say is that I think the way to make it move forward, frankly, is to provide technology that makes it easy for investigators to want to be able to adhere to the standards that people identify and to use those standards in the most meaningful way possible. I think the challenge we have right now is that creating good metadata is just incredibly onerous, and no one wants to do it. And it becomes relegated to someone who probably doesn't have a vested interest in it at all. And as a consequence, most of the metadata that people create are terrible. And unless you can provide a platform that's easy to use and guides people into using the right ontologies and the right controlled terms, we're never going to make any progress, I think. Of course, I have a biased position there, but I think it's true.
Oh, yeah. I think I would agree with that. I'm also wondering, though, if there is something that because anytime you see somebody's standard, it sounds like on the CEDAR platform, you can almost, is this correct? You can almost branch a standard from an existing standard?
Right, so you can take an existing metadata template and then edit it into your own form, retaining the fields and their associated data typing as you see fit.
Okay, and so does this capture provenance then of standards as you're doing that?
We're not, well, yes it does, because we have unique IDs for all the fields in the template. So if you look at the underlying JSON LD for the template or its instantiation, you can find out the ontologies, where they all came from. So that is true. What you don't have is provenance as one template evolves into another, which is something that we need to work on.
Because it seems like you could certainly see a consortium creating a standard that is a mesh of different standards that they need to bring in together. And certainly if you set for the entire consortium with many different data types, you're going to need to pull in standards from multiple places. And so having ways to capture kind of the provenance of standards, some places where you're going to merge them together and say, I want to use this standard for this thing, but the upstream work and the tissue processing, I need to use a standard from somewhere else. And we need to capture where these things come from.
And
right?
it can even be a many-to-many problem, because you may have a standard that acquires the metadata for some experiment, but the data themselves need to get parceled into different repositories. Sending stuff to biosample, then setting something to SRA is a good example of where that happens all the time.
And so it'd be nice if someone could put their data into this thing and say, I need to submit my metadata to HCA and to BICCN here.
Right, exactly.
Does both for me. So some of that technology would be useful. It sounds like being able to understand how the metadata maps between different consortia, which sounds like can be done in this very, very tangible way of creating these metadata templates that you guys have, and then putting this in and then doing that mapping across different metadata templates from different consortia.
Yeah, that would be great.
That's something you currently have, right?
Yeah, yeah. But I know.
Not with everything.
It'd be nice to do this, at a very large scale, and this is what the opportunity here would be.
What is it that you guys as participants are passionate about that you would like to be involved with to try to move this stuff forward so that we can get better clarity on standards across consortia, and understanding how we can set standards? I'm looking for any other feedback. I know we've got a smallish group right now.
I'd love to try some kind of proof of concept with, for example, a cross consortium or cross project attempt. So, I'd love to try our metadata specifications. So the IDR metadata specifications into Cedar, see how that works, demonstrate where we've screwed up, where effectively we're doing the same thing as others, and it's effectively test some of these concepts. With respect to the final bullet point, I can just tell you the experience we're having. So the imaging world isn't used to behaving in the way that the sequencing world is, the people on the sequencing side are used to. And so there's a bit of the answer that we have, that final bullet point, is kind of carrot and stick. The carrot is that the editor will accept your paper. So we're getting more and more submissions from authors who are just told by the editor, your paper will be published when the data is available. So there's a carrot there, but there's also a bit of a stick, and the stick is you will go through the pain of taking whatever random thing that you guys came up with in your lab and then converting it into this specification. And the only reason that we have that specification is it's what we have. I mean, it isn't as mature as any of the stuff that we're talking about. So I don't know how helpful that is or not. Ultimately, it's, in practice, people want to get their papers published and their grants funded, and that's a pretty powerful incentive.
So yeah, I'm going to say these last two bullets that we talked about, I'm going to move them down to what would encourage people to integrate with current standards. There's technology, and there's journals are cracking the whip on saying you need to have your data somewhere, and the data repositories are enforcing a standard.
Well, that would be great if it happened, but I don't.
Oh, it does happen.
Well, I mean, kind of. Kind of.
Ben, you're right. It is kind of. It is not systematic. It is not universal by any means.
Yeah, I mean, we've been pushing for journals to require standards in genomics for about 10 years now, so it's still really. I
mean, my first interaction with this was back in the mid 90s, when I was in protein structure, and it used to be people would never deposit their coordinates at all. In some cases, if they were really high profile protein structures. And it wasn't until Science and Nature refused to publish them without protein data bank deposition. And now everybody does it. But it's gotta be, I mean, you either have to, I mean, I don't know how you force a journal to do anything, right? So I think the journals are part of the problem, not part of the solution. But
well, I think the journals are turning around, because I think they recognize that they're going to get sued by people who made, well, the whole question of research integrity has really become, front and center. And I think the journals are getting nervous about this. Whether something's going to happen in our lifetime, I don't know. But I think the attitudes are changing. They're just changing slowly.
Yeah, I hope so.
I don't know if this is helpful, but back when we were all mobile, I got to have a couple drinks with Helen Berman, and she said, "Look," and we were asking me about where we were on the whole imaging thing. She said, "Yeah, you guys are like 40 years behind where we are. Right? It took us 50 years to get to this point, so it'll probably take you guys a while too." So,
I mean, the other incentive is that the whole HubMap experience is presumably going to lead to incredible data integration. At least that's the long-term goal. People will recognize that unless they apply metadata standards to their data sets, they're not going to integrate with anybody else. But that's going to be at a time when there's more data to integrate.
Benjamin, can you speak to, offhand, do you know if there are existing standards that HubMap adopted full scale, and can you tell us where HubMap is currently publishing their standards?
Oh, ENCODE, you mean?
Oh, sorry, ENCODE.
Yeah. So our standards are published on our website. They're pretty loose, though, I will say. What we do is mostly we consider our metadata standards to be part and parcel with essentially the JSON LD definition of submissions. So theoretically, one can pull it off. We have a URL called profiles that lists all the JSON objects and all their properties and what you need to submit them. It's not as discoverable as we liked. I think we adopted JSON LD so early that when Google dataset search came out, somehow the standard of LD shifted a little bit, and our data doesn't get pulled in to their system very well. So that's on the monumental list of things for us to work on. The tricky part about, I think I want to say not Anita, but no, it is Anita. Yeah, brought out is that the standards of metadata are sort of chunked out a bit, right? So if you want to talk about or Miami or whatever, or PDB is a very narrow definition of things, and Miami is actually very minimal, right? And if you look at Miami sort of evolved into sort of these, geo standards and SRA standards, and those are just key value pairs that are pretty undefined. So if you don't agree on what the keys are, and you don't agree on what the possible values are, you're not really, you're just not really doing anything. So I think that in terms of, I think what matters to all of biology is sort of how to, what if we started organizing on what is a sample, right? And you can, in the old days, this was just liver, K562 cells or something like that, right? But you could define a metadata packet that defines what a biological sample is, right? At various little types of subsamples of whatever it is that you're actually working on. And then you have to kind of go, I would say almost, I don't want to say genomics assay by genomics assay, because they are somewhat related to each other, but it's a start to just sort of do something small at first. It's a very huge area to try to map across, whatever people are doing, or I mean, if people are all working on single cell RNA, single cell attack, try to figure out what, if you know what experiments people are doing and what they're going to do in the next 4 years, try to determine what are the possible ways to fractionate that data and how people might search it, and then work backward from there. I don't know if that helps or not, or answers your question.
I feel like that's going into a lot of detail on the specific fields. I guess a lot of my trouble is just even getting to the point of understanding what other people are doing. And so if I wanted to harmonize with anybody, how do we do it? And then different groups will prioritize working on harmonization or definition of different sets of metadata at a time. And so trying to understand how doing, so perhaps you're saying that, I mean, there's the getting agreement on the actual metadata schemas for different aspects, whether it's samples or whether it's transcriptomics analysis or something, or whatever it is. It does seem like it would be useful to have very specific focus groups on those things. So that first step is just documenting what did you make? Where did your fields come from when you did this? It didn't come from out of thin air for the most part. A lot of times people are opening up different things and saying, we'll take a little bit of this and a little bit of that. Here's the HCA standard. Here's what's an SRA, which is much smaller. So let's see. Okay, I'm trying to figure out what is the best use of the next, what do we have, another 10 to 20 minutes, I think, left in the session. So for coming up with actionable items, coming up. I guess I should also say, Benjamin, there's a separate group happening in parallel right now on common metadata schemas.
Yeah.
And
We have people, I have multiple people in that one. We divided up probably 6 people across 4 sessions in this. So I got the first pick, so I'm here.
So they're working, I feel like more on some of the specific attributes. So that's why I'm kind of also trying to keep the session more on how do we deal with the federation and of different groups.
Well, I think what that I'm not sure, the idea of not worrying too much about, like, there's an internal representation and an external representation, okay, right? Like it's, you can't, especially if you're in a, like, I mean, maybe we were in a strange case because we became the data coordination center of a consortium that had been in existence for 6 years, right? So there wasn't like this spin up period of like, oh, what are we going to do? So, you know, we had to sort of jump on the horse as it was running by. And I think that my presumption is that a lot of people are in this situation where work is being done and the data coordination groups are then trying to figure out how to model the data that's coming out. And I think that that's not something that you can apply a standard to, right? You just have to sort of catch up with what you're doing. And then the federated part of it, the federated metadata standards it has to do with, you know, what do our groups have in common, less than what standards exist. Like the standards that you're going to need in your project, whoever anybody is going to vary like tremendously, in the sense that like I mean, you're just going to have to start somewhere and build something up. Like, but again, that goes back to the other group. In terms of the federated stuff, like, I don't know that, like, I think it's hard to, I think it's backwards to look at it that way, to try to come up, to look at the standards and then say what you need to do, unless you have no idea. But
well, I guess I'm looking at this in terms of there are a number of standards that exist. So ideally, we're not starting from scratch everywhere. In practice, a lot of us start into this and you have to start with something. And so you start from scratch and then you try to catch up to what should be your best practice, what are the standards that are out there. But there are a lot of new groups that are funded to set standards. And so this is, and I'll say it's also challenging because it's not like we have one data modality is at one data archive. So we've got NEMO, who's collecting transcriptomic data for the BICCN. And I think there are some other consortia you guys may be affiliated with as well, right? But there's also transcriptomic data at the HCA, and there's transcriptomic data at HubMap. And so we have different archives involved in different consortia, each setting their own standards. Whereas we still live in this space where we could, it's the same data generating labs for a lot of these. We should be playing in the same space of coordinating on some of these standards. At the same time, there may be other genomics or transcriptomic standards being set by totally other groups out there as well. So this is where it's what little steps can we take to help people, help consortia get up and running and have some standardization. Yes, we all have to do things that make sense for our own consortium. We're building different back-end things that require different types of information. Some groups may be able to suck up a lot of information. Other groups only want a very minimal amount because that's what they're willing to curate and deal with. But what can we do that helps everybody, right? So I think this is where we're trying to make some steps forward or figure out a path forward.
And I guess it's not that hard in some sense, because even though each group may view itself very specially and you have particular goals in mind, there are only a handful of stereotypic experiments that people tend to do. Well, maybe not a handful, but a finite number. And I think if we can identify through some mechanisms for those particular kinds of experiments that right now don't have minimal information models or any kind of existing metadata standards, that's a place to begin, and to begin to flesh out the landscape to make sure that we can have standardized things to say about the kinds of experiments that really matter. Yes, I mean, there may be arcane things you may want to say about experimental conditions, depending on what the experiment actually is. But there's usually some very basic stuff that's going to be consistent across experimental types, and those can be standardized.
I think use of for something that's actionable to help answer this question, some specific examples might help people in general. In my personal experience in working with pharmaceutical companies and looking at the clinical data side, standards are great, but it also is up to the individual that is interpreting the definition of the standards themselves. So, like, to give one specific example, I've worked with, randomized controlled trial data standardized into submission format by same data set by two different groups, and the output that I get is completely different because of the way the different groups interpret the information. Now, it might be different for more omics or molecular type of information where the assays might be standardized and the information might come out of instruments. But these are things that, there's a level, I think just looking at standards by themselves without looking at how the information is coming into the standards might cause trouble.
Anita, I agree with that completely. And I think, frankly, the problem of getting people to apply standards in a reasonable way is probably a bigger problem than coming up with those standards in the first place. We've done a look at metadata that exists in NCBI, and it's unbelievable how bad it is. We looked at biosample, and like 76% of the Boolean values are neither true nor false, just to give you a flavor for how bad people apply standards. Like 26% of the integers are not parsable as numbers. So this is the kind of stuff that people do when they're left to their own devices. And I think you're right. This is probably a bigger challenge than coming up with the standards in the first place.
Yeah, and I don't think that your experience is, Anita, is specific to clinical samples. I think one of the issues that what you're saying, and this goes back to what Mark is saying about geos specifically, is that there is a standard. It's just because it starts from a place of like non-initiating work, it's essentially impossible to update old data to current standards. And so people end up writing a standard for a repository like GEO, which is meant to contain essentially every genomics experiment ever done, paid for by NIH. The standards themselves are just not specific enough to be interpretable. I don't know if that solves any problem. This is causing more problems, but I'm not actually helping you with action items here. The action item is really tough. It's just it's a lot of work to do this. It's a lot of work to have people, collate and cross standards and try to figure out why. It's just the newer groups have it easier though. The newer your group is, the better you can learn from everyone else's attempts and sort of pick, which one fits you the best. And then like if everyone's picking the best one, and I think that as Jason said, like if people started using common software, I know there's a product out of University of Chicago called Gen 3, which is a very generic like data coordination center piece of software, which has a lot of good features. If like people start using that, like they correspond a little bit easier. There's 4 or 5 consortia that use our software on the back end, but the whole point of our software is that the schemas are flexible. So, you can adjust the schemas to fit your product.
So is everybody looking at the little blue box on the Google Doc? So to try to finish this up, we wanted to set a playbook for setting standards, finding and integrating with them. And so that first step is that first bullet point we have on here, that NIH consortia join a workshop on finding and sharing standards. To finding solutions to that. That could also be a workshop if such a thing were held that would talk about kind of what there's finding and sharing standards, and then that could potentially there could be a way to talk about best practices for setting standards, and maybe that would be something useful there could be something like retrospectives on having set standards by different groups, kind of successes and failures of different things. Does that sound like something that we could put together into a workshop? Maybe that could be something at, you know, if we had if there were another NIH-HCA meeting or Brain Initiative investigators meeting or around SFN or something. This would be, I mean, this would be asking NIH to help coordinate something like this. Would this be a big thing or would this be a small workshop? What would this look like?
Well, I'm thinking about 6 years ago, when BD2K was first taking off, NIH did a very big workshop on community-based standards, including metadata standards. But that was more for them to identify what is the landscape of standards rather than to actually roll up sleeves and figure out how to create one. So there's at least there's precedent for NIH caring about this kind of stuff. I think the goal here would make it a little more focused.
How would you phrase the goal for this workshop?
To articulate mechanisms by which consortia of investigators can learn from one another's metadata. I don't call them, they're not standards yet because no one, metadata contributions, and to work towards standards that can be applicable across a broader community. How's that? Did anybody catch that?
It's being written for you right there.
Oh, okay. Well, I don't have to change windows to see with the Google Doc.
It appears by magic. Thank you, Kylee.
It really is incredible. Harry Potter.
I've given up. I can't keep up with Kylee.
I feel like I can keep up with all of you, so don't worry.
I feel like that's a good articulation of that. The one thing that I would add there, of course, is the understanding what standards are in use.
Yeah.
By the different already in use. Like, so standards not necessarily that they were creating, but how they're interacting with existing standards is one addition.
Yeah, I wonder if it'd be nice to have kind of a practical component to it. So pick, you know, a digestible number of proofs of concepts where the various consortia make an effort. And this, as you guys have said, this has already happened in some sense to adopt or connect with standards. How did that go? What needed to be changed, et cetera? How did they deal with the inevitable friction points, or, you know, or car crashes or whatever they are.
Where did a consortium or a standards effort end up finding that they have to do rework? And what caused the rework?
Yeah, and how do you do rework? It took us a while to understand that actually when there's a whole bunch of ontologies we're using in IDR, the right thing to do is to call EBI. You know, we call Helen Parkinson or call somebody in her team and say, right, this is what we need to do, as opposed to do the work ourselves. Because actually, it wasn't so much, it's not a question of delegation. It's a question of understanding what someone else was trying to do, how far they got, what they understand the limitations are, and then working together to kind of that up. So that, you know, inevitably what we ended up discovering is somebody had some ideas that we were thinking about. You know, that goes back to the phone call thing. I know I'm just repeating myself here. But,
okay. Does anybody have anything else? I feel like we've kind of reached a rough draft of something that would be useful to have at some point. And so then this would be a discussion with NIH and then finding some good ringleaders to help rope all of this together. But this seems like something to propose to the NIH as a follow-up that would be useful.
Sounds good to me. It's very concrete.
Does anybody want to be a ringleader? Everybody will participate.
I'm happy to help out since I have no vested community that I'm adherent to.
Okay. Well, I think we'll assume everybody would like to participate, and then maybe we can put as a proposal to NIH and then find some good ringleaders from that across the different consortia as well to help put something like that together. This seems like this would have been a useful adjunct meeting to the NIH-HCA meeting if it had it been in person or something similar in the future.
So maybe in 2021. Sorry, I should not even joke.
Do we want to remove, there was a note above about like doing some testing of technologies that make it easier for people or to help people submit across different consortia. Do we want that to be one of the recommendations? And I think
that's something this group, I think you're right. Thanks for bringing that back up. Jason had said he was going to try to take the IDR and put that into the CEDAR platform. I haven't looked at CEDAR yet. I would love to look at CEDAR and see what's there. I'm not sure what I would put into it yet, but would be interested in trying it out. We're trying to develop some standards, so as we have something that we can put in there, we can try. Like Heather, we can probably do something there on the transcriptomic side too, right? Take a look. But I need to look at it and see what we might be able to put in.
Yeah, maybe a question is for those pieces of work. Where do we report back? And I mean, obviously, I'm definitely going to talk to Mark, but it'd be interesting, is it this group or whatever?
Are you guys interested in meeting again in, say, a month after taking a look at the CEDAR thing? I think we could meet. I'm not quite sure how to follow up on the workshop piece with NIH. I think I would start with Yang Yao and Ming Zhan, who are the program officers that I'm familiar with at NIH, to talk about how something like this might happen. That would be where I would start. Does that sound good?
That sounds good. And if looking at the website is not enough, or just getting an account and playing with it is not enough, we're happy to do a demo or something like that.
That would be awesome. We could try to schedule a demo and invite a large group of an audience from across the NIH-HCA, just as they were holding the pre-meetings before this one. Perhaps we could have some follow-up meetings that people are invited to about how to use CEDAR.
Be happy to do that.
I'd be interested in attending for sure. And then we could, if we try that out, we could try to schedule a follow-up call with, we could invite everybody here, and then those of us who might try to put some stuff into IDR into CEDAR could try to do that. Kylee, as a representative of HCA, do you think HCA, is there someone at HCA who would take a look at that?
There are definitely folks more familiar with this topic at HCA that I can connect you to, or invite them to this if you'd like.
So I would say maybe we can try to do a follow-up in about, I'd say a month. But ahead of that, I'll contact program officers about the consortium and see what response we get back. For looking at CEDAR, we'll also ask them about whether we can have a demo to the broader NIH-HCA meeting audience about using CEDAR. I think that would be awesome.